We are Justin's co-advisors, which is why both Michael and I are here. And frankly, it took two of us, it's good he has co-advisors, just to keep up with Justin. Justin did his undergrad at UCSB, where he also worked for the Cheadle Center for Biodiversity and Restoration. And I see we have some representatives from there today. He arrived here in September 2017. He also started writing grants and collecting data for his dissertation in September 2017, and he has not slowed down since. It's now 4.5 years later, and he has received more than 25 grants for his research. He has also, during his time here, published 6 papers, and that includes last week, he actually had his first solo author paper accepted, and he now has 3 more papers in review and several more in the works.
Justin, if you're not aware of it, he's an exceptional mentor, and he has included almost 230 high school and undergraduate students working on his research. There's a whole subculture of ImageJ experts running around out there, including 15 independent research students, but these numbers don't tell the entire story. Justin works really hard to feel that students feel included and grow academically from the research experience. Justin wrote several, I think 6 postdoc proposals, one of which has been recommended for funding. We're not allowed to say much more than that, to work with Jen Fung at UC Davis. And we're really proud of everything that Justin has accomplished, but we're of course sad to see him go, as he's going to be leaving a gigantic hole in our labs.
And with that, we'll pass it over to Justin. Congratulations.
Thank you, Michael and Karen, for that kind introduction. And thank you everyone for joining me today for my exit talk. I see a lot of familiar faces, and it's really great to see everyone. And without further ado, I'll be talking about my dissertation and my work over the past 5 years. And my talk today is called Assessing Regional Outcomes and Drought Adaptations for Coastal California Grassland Restoration. Before we get started, I just want to do a few acknowledgements. The first one is a land acknowledgement. And I just want to state that the research that the land on which my research was taken was the unceded territory of numerous indigenous tribes which were violently removed from their lands, which included but are not limited to the Chumash, Selenian, Amamutsin, Ohlone, Miwok, Pomo, Wiyot, and Wiyot tribes, and are today working towards decolonization and traditional land stewardship. I also want to thank my committee and my PIs and various lab groups that have helped me throughout the years, and the numerous restoration practitioners that have provided input and helped me find research sites, and many other faculty, and also the wonderful artists that have worked with my work, which I'll be referencing throughout the talk. I also want to thank my numerous field assistants and student interns, my many generous funding supports, and my Patrick Turner, my husband, and my parents, and the rest of my family. Before we get started, I just want to do a quick overview of the talk today. So as we go through the talk, we'll first discuss the background information, then my first chapter about plant-based, trait-based plant selection for climate change, and then thinking about how plants balance trait development for drought and competition, and then finally zooming out and thinking about how successful are current restoration projects. As we progress through each of these sections, you'll notice one of these little icons in the upper right-hand corner of the screen, which will indicate what section of the talk we're in. Restoration is a growing priority, both locally and globally. Increasingly, there are interdisciplinary stakeholder meetings that result in initiatives calling for the protection and restoration of biodiversity. For example, internationally, we have the Convention on Biological Diversity, which helps set global goals for biodiversity protection. More recently, the UN also declared 2020 a decade for ecological restoration, with ambitious goals like the 30 by 30 initiative, which seeks to conserve 30% of Earth's surface by 2030. More locally, we have the California Coastal Act, which was passed in 1976 and mandates the restoration of coastal areas that are affected by coastal development. We also have the California Biodiversity Initiative, passed in 2018, which places further emphasis on protecting native California flora, and fauna. Past studies have even shown that local community members are willing to pay into restoration as long as that work is going to occur within their local community and not elsewhere in the region. However, there is some uncertainty with restoration currently because outcomes are relatively unknown and can sometimes be variable. On the photos on the right, you can see 3 different transects I completed at a single restoration site. Even from this one site, you can see how much variability there is in outcomes. Most grassland restoration projects also lack funds to conduct post-implementation monitoring, which prevents them from knowing if they achieved their project goals. This also means that many projects are not able to apply adaptive management because there's insufficient data about their previous work. Projects that do have funds often are only able to monitor immediately after implementation or for a short duration thereafter, making outcomes of projects past 5 years relatively unknown. Experimental restoration studies in a variety of ecosystems also indicate that the wide range of variability may be caused by environmental stochasticity or randomness, limited resources, and insufficient knowledge about specific local natural history of rarer species. Ecological restoration also requires direct human intervention, and as such will be shaped by the person or persons undertaking the restoration. It has been shown in various studies that socioeconomic and political factors play a defining role in shaping ecological outcomes, and even more so in projects with direct human intervention. For example, the surrounding parcels and their views and perspectives may influence how an agency undertakes restoration, or may even limit the tools practitioners can use, such as prescribed fires or herbicide. On the other hand, a land manager's interest may drive or create restoration projects where uninterested people might decide to pursue other types of conservation projects. Somewhere that has access to machinery from nearby agricultural parcels may use similar practices as nearby farmers, whereas agencies with high staff and volunteer labor may be more often use large groups that do hand removal. Some may even have different species preferences or different landscape aesthetics. As such, management practices greatly differ across restoration agencies and even within the same agency across different practitioners. As such, restoration project goals also often differ. Goals may also differ depending on whether or not the project is a statutory project, meaning it's legally mandated by law, or if it's a non-statutory project, meaning it's not legally mandated and essentially voluntary. However, the commonality that does continue to tie together most restoration projects is that they are often fund limited. It's also hard to address the variability in restoration because of a critical science practice gap. This gap leads to research and management practice that are not always synergistic. This lack of communication leads to experiments that may not be applied as real-world solutions because they don't readily match management problems or local environments. This gap is also facilitated by differing goals and resource levels in restoration research versus practice. For example, research can generate expensive, labor-intensive suggestions that are not always feasible for implementation or practice. Furthermore, research is often focused in theoretical frameworks that are technical and not always apparently relevant for land managers. The gap is also exacerbated by hard-to-access published research, which is often behind paywalls if not directly shared with practitioners. Research has suggested that functional traits could be one way to address variability in restoration site conditions and project outcomes because quantitative trait values can be matched with environmental conditions and plant establishment. In theory, changes in precipitation could cause changes in environmental filters and affect the spread of values in traits and leave a greater proportion of remaining individuals in the community with functional trait values conferring drought resistance. Environmental theory then predicts that the individuals that pass through the abiotic filter must pass through a biotic filter, which can consist of factors such as competition, disease, or other related biological factors. Resulting communities will then be a subset of regionally present taxa to have trait values that balance the local abiotic and biotic site condition. On the slide, we have a conceptual figure to represent this kind of theory. So each shape represents an individual taxa, which you can think of as a plant or animal or anything. And then there's an environmental filter like drought or specific soil conditions that only certain species or taxa can pass through. And so here we see a subset of those. And then those species may compete with each other or experience disease and have to pass through a different set of filters. And the remaining species that are present in that local species pool are a subset that can balance those stressors. Furthermore, given future climate change, optimal partitioning theory, which within plant physiology predicts that plants will allocate biomass to structures that increase uptake of the most limiting resources. For example, strong abiotic filters may lead plants to allocate more growth to below ground structures, but less is known about how optimal partitioning balances biotic filters. For example, competition could increase above ground allocation to compete for space and light, whereas it could also increase below ground allocation if resources such as nutrients and water become more limiting due to the invaders. Mechanistic understanding of trade-offs in trait development could help inform restoration plant selection. Although there are numerous functional traits that can be assessed, here are a subset of a few important traits that I'll often refer to throughout the presentation. So first we have leaf lobeness, and similar to how humans can cool through sweating, plants can also cool through transpiration and water loss. However, increased leaf lobeness allows for a plant to cool instead using nearby air and reduce the amount of transpiration, or in a sense, sweating, to cool. Specific leaf area is related to the amount of investment into a particular leaf, and the higher the leaf, the higher the specific leaf area, the lower the leaf lifespan. And specific leaf area is also related to other things like drought resistance and herbivore resistance. Major vein length per unit area is related to the amount of water and sugar transport a leaf can support. Water use efficiency is the relationship between carbon assimilation, which you can also think about as the photosynthetic rate of plants, how much carbon dioxide is taken in by the leaves to be made into sugar, over as a ratio to stomatal conductance, which is the amount of water leaving the leaves. And so water use efficiency is essentially how efficient a plant is in taking in water without losing too much, taking in carbon without losing too much water. We also assessed growth rates, and quicker growing plants are often thought to be more resource acquisitive and have different growth strategies, whereas slower growing plants often are thought to be more conservative. Overall biomass can also be used to assess relative allocation to stressors. Phylogenetic diversity has also been suggested as a quantitative method to improve restoration establishment and decrease variability in project outcomes. Broadly speaking, phylogenetics is the evolutionary relationships between taxa measured in the divergence time of millions of years ago, typically represented through a phylogeny. Density dependence theory predicts that species that are closely related are more likely to compete for similar resources and share diseases, limiting the abundance of related taxa. Furthermore, phylogenetic niche conservatism predicts that closely related species are also more likely to share similar trait values and thereby more likely to have similar outcomes in restoration planting. Phylogenetic relationships are often assessed through nearest taxon distance and mean phylogenetic distance. To better understand the long-term outcomes of restoration projects and the impacts of future climate change on restoration methods, I focused on California coastal grasslands. I focused on coastal grasslands because they're a small sliver of the 1% of remaining grasslands in California that are unique and one of the most diverse grassland types in North America. In part, high diversity is due to topographic diversity of California and summertime fog, which contributes to plant water use in the dry season. High diversity is also often attributed to high annual forage richness, but it's often hard to restore, which is leading to something we call the perennialization of California grasslands, which is the increased dominance and abundance of perennial species. Coastal prairies are increasingly also affected by land development, which can directly replace habitat because many people want to live by the ocean and beach. And it can also indirectly contribute to habitat fragmentation, change disturbance patterns, and facilitate species invasions. Because coastal grasslands are often located within a legally designated coastal zone, they often require mandated restoration through the California Coastal Commission or by county general plans. So now that we've covered some of the background material, we're going to move on to my first chapter, which thinks a little bit more about trait-based plant selection. And this chapter has also been published, so if you want to read more about it, you can find more details online at the Journal of Applied Ecology, or you can also email me for a PDF copy. In order to understand the utility of trait-based selection, I designed an experiment on an invaded coastal prairie at Younger Lagoon Reserve using the International Drought Experiment design, which is pictured in the bottom left. These plots are 4 by 4 meters, and the drought structures are designed to exclude 60% of incoming rainfall, simulating a 1 in 100 year drought, which are also confirmed with soil moisture probes. Control plots, which are pictured here on the bottom right, received ambient rainfall conditions, which means the weather wasn't manipulated on those plots. Each plot was planted with several individuals of 12 different native species, and then weeded twice in the first year and received no management thereafter. With this design, I asked whether or not drought-related functional traits and phylogenetic relationships were good predictors of survival. To answer these questions, I used a standard protocol to collect functional traits such as growth rates, specific leaf area, leaf lobeness, leaf carbon nitrogen content, and delta carbon 13, a proxy for water use efficiency. I measured the absolute plant cover of all plant species in each plot using 6 quadrats. Plant communities were then used to create phylogenies and also summed to assess native and non-native plant cover. Plant cover was also used to abundance weight traits and phylogenetics for community level analyses. We found that drought affects restoration and that traits help explain the differing plant communities. With an analysis of variance, which is used to compare the means of average plant, which was used to compare the means of average plant covers between plots, we found that droughted plots often had higher native cover and lower non-native cover, which is represented in the box plots here. So on the on the x-axis here, which is not does not have words, it's instead represented through color. We have ambient rainfall in green and drought in red. On the y-axis, respectively, we have native plant cover and non-native plant cover. And as you can see here, native plant cover is often higher on the drought plots and is lower, and non-native plant cover is lower on the drought plots. We also found, using a Bray-Curtis dissimilarity index and analyses, that experimental treatments resulted in differing plant communities, and that higher trait values of certain traits like lobeness and C and carbon nitrogen were associated with key treatments. This was represented through a non-metric dimensional space plot, also known as an MMDS. In the NMDS, each point represents the average plant community of a plot in a given year. The ellipses represent the average space of plots in a treatment group or sampling year. Therefore, the MMDS here shows that there were differences in drought and ambient rainfall plant communities while accounting for interannual differences between 2018 and 2019. A permutational analysis of variance also showed that plants on drought plots often had higher leaf lobeness, while plants on ambient rainfall plots often had higher leaf carbon nitrogen, indicated by the vectors in the MMDS. On these next series of figures, I'll be discussing the mortality risk of native plants with different trait values. Each row represents a trait or phylogenetic metric, and the dashed vertical line represents the null axis. If a point or its confidence interval crosses the null axis, that trait is not a significant predictor. If a point does not cross the null axis, higher values of that trait represents the multiplicative mortality risk associated with the hazard ratio on the x-axis. Overall, we found that increased leaf lobeness decreased mortality risk, whereas increased growth rates increased the mortality risk, regardless of treatment. For plants on drought plots, we also find that increased delta carbon 13, a proxy for water use efficiency, and the increased nearest taxon distance increased mortality, which were not significant for plants on ambient rainfall plots. We found that intensifying abiotic conditions will promote functional traits that reduce water use and to survive intensifying abiotic filter when establishing in restoration communities. We found leaf lobedness and slower growth rates increased water use efficiency were important for survival during drought. Highest nearest taxon distance was related to increased mortality during drought, which means plants less related to the surrounding plant community were more likely to die. Although this is inconsistent with density dependence theory, this could be because leaf traits were conserved, meaning they were similar, allowing plants in the communities with similar traits to survive the same intensified abiotic filter. And this was further supported by trait conservatism analyses for leaf lobedness and delta carbon 13. These results also have management implications. First, drought will affect plant community composition and may indicate management practices could be adjusted. We found that there was greater native cover and lower non-native cover on drought plots, indicating that native species may be experiencing competitive release. This may also suggest that management may be able to shift some resources from invasive control to other actions during drier years. We also found some traits were more important than others, but there are still management limitations to implementing traits because it's very labor intensive and the data is not widely available for all species and traits. However, this is slowly changing with new platforms that are trying to integrate trait modeling and data archives. The next project I undertook was because I saw the potential effects of competitive release of native species in the field in response to drought. However, because there's so many different confounding factors in the field, I tested this in a controlled greenhouse environment to better understand mechanisms of trade-offs for drought and invasive competition. To address the field observations I saw, I asked, how do common restoration species balance drought and invasive competition stress? And how do they experience competitive release from non-native species during drought? I used a full factorial experiment design with drought and invasive competition in a greenhouse. We implemented episodic drought where plants are withheld a minimum stomatal conductance withheld water until they reach a minimum leaf stomatal conductance threshold. And just a reminder, stomatal conductance is the rate of water leaving the leaves. And when the plant reaches that minimum threshold, we can essentially consider the stomata closed and the plant drought stressed. So after the plants reach that minimum stomatal conductance threshold, they're rewatered and allowed to be rewatered for a few days. And then they experience drought again, and final measurements are taken once they reach the stomatal conductance threshold during the second drought period. Competition was induced through sowing 5 non-native species that were dominant from the field plant surveys and used at densities equivalent to field abundances. I collected above ground and below ground biomass and leaf traits prior to treatments to establish a baseline, and at the end of the treatments to compare the different effects. I also measured leaf carbon assimilation, also known as photosynthesis, stomatal conductance, which again is the rate of water leaving the leaves, and the water use efficiency, using an infrared gas analyzer weekly during peak daylight hours and once at the end of the treatment overnight. For the purposes of this talk, you can essentially consider carbon assimilation and photosynthesis to be the same thing. I also calculated various metrics of photosynthetic recovery to assess drought stress with and without invasive competition, which I'll briefly explain with this conceptual figure. So first, I want to point out there's 3 different terms we're thinking about. The first is PLA, the percent loss of assimilation or photosynthesis, ARR, assimilation or photosynthetic recovery rate, and PRA, the percent recovery of assimilation or photosynthesis. On the x-axis, we have time, and on the y-axis, we have A, which stands for carbon assimilation or photosynthesis. You can imagine at a starting point, a plant has a baseline photosynthetic rate. And then a stressor comes in, and in our case, it's drought, and the plant loses some of its photosynthetic ability and photosynthesizes at a lower rate. And this is what we call the PLA, or the percent loss in assimilation or photosynthesis. After that stressor is removed, and in this case, that's with rewatering, we can see that the plants sometimes recover their photosynthetic ability to the same level or sometimes a lower level. And this amount of recovery is considered the PRA, or the percent recovery of assimilation or photosynthesis. And then finally, we have ARR, or photosynthetic recovery rate. And this is the rate, or how quickly the photosynthesis is recovering for the plant after the stressor is removed. Although we found a lot of cool stuff during this project, I'm just going to go over a few key highlights today. First, we found that certain species are more likely to lose photosynthetic ability from drought, as we can see in this first graph. In this first graph, we have PLA, the percent loss of assimilation or photosynthesis on the x-axis, and we have the 5 different native species we worked with on the y-axis. For Bromus carinatus and Lupinus nanus, you can see that there's greater percent loss of assimilation when in invasive competition, which is expected. But for Diplodocus aurantiacus, we actually find that it has lower percent loss of assimilation when experiencing invasive competition, which could potentially support competitive release. We also found that some species recovered faster when in competition than others. So in this case, we found Bromus carinatus actually, on the x-axis for this figure, we have ARR, which is the recovery rate of photosynthetic ability. And we find that Bromus carinatus actually has quicker recovery when experiencing competition, which may also indicate that it is experiencing competitive release. Diplodocus aurantiacus also had higher water use efficiency when experiencing both drought and competition simultaneously, but not individually. And that can also further support competitive release. These two species, Bromus carinatus and Diplodocus aurantiacus, were maybe experiencing competitive release because they were not able to adjust their root to shoot ratios in the competition treatment. We also found that physiological and morphological traits fell along 2 key axes that were partitioned more by species identity than by treatment. On this figure, each point represents the average trait space occupied by a particular individual. Colors represent different plant species, and shapes represents different experimental treatments. The first axes represented different types of resource management strategies, such as acquisitive growth with high growth rates, photosynthetic rates, and conservative growth, and on the other end with high VLA, major vein length, and leaf slopeness. The other axis was related to above ground allocation and below ground allocation. Overall, we found that most species we studied exhibited drought-adapted trait adjustments, and less so for competition. Results supported that Bromus carinatus and Diplodocus aurantiacus may experience competitive release during drought. Consistent with environmental filter theory and optimal partitioning theory respectively, we found trade-offs between leaf traits conservatism versus acquisition and below ground versus above ground resource allocation. In both instances, trade-offs appear to relate more to abiotic stressors of drought compared to biotic stressors of competition. Although above ground allocation may be a method for adjusting for drought, we found that the species that drove this trade-off was Diplodocus aurantiacus, which also develops woody tissue, and which is another potential way to facilitate drought resistance. So now that we know a little bit more about how plant traits can inform restoration plant selection for future droughts and how plants may develop trade-offs in their traits in response to competition and drought, we can zoom out a little bit more and think about how current restoration projects are completed and how their outcomes are faring relative to their own project goals and a standard performance metric for native cover. To better understand coastal grassland restoration outcomes years later after implementation, I asked 3 key questions. Does coastal grassland restoration meet project-based goals and a standard performance metric for native cover? Is native cover related to project age? And what are the biggest barriers to achieving restoration goals? To answer these questions, I identified 37 restoration projects spanning along a 1,000 kilometer north-south climate gradient from Santa Barbara to Humboldt counties. To locate projects, I first contacted universities and colleges in each of these areas to see if they knew of any restoration projects. I also contacted land trusts, nonprofits, local nature reserves to see if they had completed any restoration themselves or knew of any nearby projects. I then reached out to my known contacts in restoration from these areas and by identifying local restoration agencies. Initially, I contacted about 150 people from the search, which led me to make new contacts and eventually talk to around 200 people to identify sites. I later learned, after my initial surveys in 2019 from interviews, that there may be up to 48 sites that meet my criteria, indicating that I likely have surveyed about 2/3 of the known coastal grassland restoration projects within this range. Sites selected were at least 3 years post-implementation in 2019. They were actively planted or seeded, larger than 1 acre in size, and received summertime fog. I selected projects 3 years or older because first-year results can often vary widely because sometimes plants experience high mortality in their first year, or in other cases, grow slow and don't achieve higher cover until later years. The projects I identified ranged from 3 to 30 years post-implementation by 2019 and ranged from 1 to 32 acres. I conducted vegetation surveys in 2019, 2020, and 2021. In 2019, I surveyed the original 32 sites I identified, and then afterwards I conducted interviews with practitioners to find more. In 2020, I couldn't gain access to all the sites due to COVID-19 restrictions and timing, and sampled about 20 sites. In 2021, I resurveyed most of the original sites and 5 additional new projects from the management interviews, totaling 34 sites for that year. I estimated that absolute plant cover of all plant species and ground cover types in 1/4 meter square quadrats every 5 meters along 50-meter transects. I used a minimum of 3 and a maximum of 16 transects, depending on the site size. In 2019, I also collected soil samples to characterize the site soils. Before conducting the vegetative surveys, I also conducted project document analyses to assess the original project goals, resource levels, and plant selection. Only 11 of the 37 projects I worked with had project documents, but often did not have all the data I needed. After conducting the vegetation surveys in 2019, I conducted semi-structured interviews with at least 1 restoration practitioner from each project. Semi-structured interviews have guiding questions but are loosely structured so practitioners can lead the discussion. Because some practitioners worked on multiple projects I surveyed, they may be linked to more than 1 project, thus there were less than 37 interviews. When possible, I interviewed 2 people from each project and in total interviewed 26 practitioners. Interviews focused on the largest barriers to achieving restoration goals, implementation strategies, and helped fill in information for projects that had no documents or missing information within their documents. It's also important to note that when using mixed qualitative and quantitative methods, that qualitative social methods cannot always be analyzed in the same way quantitative ecological data might be. And in this project, qualitative social methods were used to help to provide context and help explain some of the findings we're seeing with the vegetation surveys. I also want to mention before we get more into these results that the data I'm presenting for this chapter is only from 2019 and I'm still working to incorporate 2021. We found that all sites but 1 were successful in reaching their project-based goals. And we also found that restoration was largely successful at reaching a standard performance metric. The standard performance metric we set for this project is 25% native cover and 6 native species, because that's similar to some targets that compliance projects have, and because it's often really hard to keep high native cover in grassland systems, which have extreme difficulty with species invasions. The graph on the right shows whether or not a project, or shows the relative proportion of projects that met the standard performance metric. We find that 81% of the projects indicated by this dark bar met the standard performance metric of 25% native cover and at least 6 native species after 5 years. With the light tan bar indicates that 6% of projects met the species richness goal, but fell 5% short of the native cover goal. And finally, the tan bar indicates that 13% of the project survey need further improvement to meet the standard performance metric. Another interesting finding that we had was that voluntary sites, which were ones that were not legally mandated, often were found to have greater native species richness, whereas statutory projects, which were legally mandated by law, often had lower non-native cover, but also had lower native species richness. It's also worth mentioning that even though I had an equal proportion of voluntary and mandatory sites surveyed in this project, there were about 9 additional statutory projects, which are those that are legally mandated, that denied, specifically asked that I did not come to their property to assess the restoration project, even though I told them that the data would be kept anonymous. We found that labor investment affects restoration outcomes. Specifically, we found increased labor can help projects achieve higher species richness and lower non-native plant cover, as indicated by these box plots. In these box plots, the x-axis represents labor investment from low, medium, and high. The y-axis represents, respectively, native species richness and non-native plant cover. We find for native species richness, medium and high levels of labor investment help improve native species richness compared to low levels of labor investment. For native plant cover, we find that increasing levels of labor investment decrease non-native plant cover, or are related to decreased non-native plant cover at these restoration projects. We found that monetary or funding values or funding resources were not directly related or correlated to native and non-native plant metrics, potentially because funding was really variable and in some cases inflated for other costs like construction. For example, one of the projects was a former lumber mill. So a lot of the costs there, millions of dollars, went to ripping up asphalt, bringing in new soil. And so not all of that work was directly used on what some people might consider as direct restoration activities like weeding and planting and seeding. Now, before we get into this next section, I just want to state I will be presenting a series of correlation plots. And it's important to remember that these variables presented may be related or correlated, but that doesn't mean that one caused the other. We found that native species cover ranged from 13 to 79% across the sites that I surveyed, and native species richness ranged from 5 to 60. We also found that native species cover did not change strongly over time, with no significant correlation or decline in native cover with post-implementation age. We found that non-native cover ranged from 24% to 96%, and non-native species richness ranged from 15 to 47 species. We did, however, find a weak 9% correlation between post-implementation project age and non-native species cover, meaning that increased non-native species cover is related to increased post-implementation age. Furthermore, we also found there was a strong negative correlation between non-native plant cover and native plant cover, illustrating potentially the difficulty in invasive species management. We also found that non-native plant cover was also negatively correlated to native species richness, which is not pictured on this slide. Practitioners from all projects indicated that invasive species management was the largest barrier to achieving project goals, further demonstrating difficulty in managing invasive species during restoration. We found that 84% of the projects indicated that they had insufficient funding, or it was a barrier to achieving the quality they wished to achieve. From the 27 projects that indicated they had insufficient funding, 74% of the projects indicated they would have conducted post-implementation monitoring if they had sufficient resources, and they would have also tried to apply adaptive management. 33% of the projects also indicated they had difficulty in sourcing appropriate plant material due to insufficient knowledge about natural history and where to collect these species, how to propagate them, and sometimes through a restriction in planting genetics. One of the really important and interesting findings from this study was that coastal prairie restoration may be causing regional biotic homogenization. Biotic homogenization, simply put, for those that are unfamiliar, is the reduction of biological diversity through dominance of a few key species. Surveys indicated that 88% of restoration practitioners across the entire 1,000 kilometer span of restored coastal grasslands are using 8 key species because they believe they will either have better survival or grow faster to meet project goals, which makes sense because obviously no one wants to fail at meeting their project goals or their personal goals. In other words, at a regional scale, species may be becoming more similar. In this table, we can see the frequency each species is used across the 32 projects, where the green N columns represent northern sites north of the Golden Gate Bridge. The C blue sites represent Central Coast sites south of the Golden Gate Bridge, but north of San Luis Obispo, and orange represents southern sites in San Luis Obispo and Santa Barbara. It's notable that all species commonly used were perennial grass species, with the exception of Achillea millefolium, which is a circumboreal forb found in many other types of habitats. Yeah. 3 of these species were also used in 50% of all of the sites across the 1,000 kilometer range. This may also be contributing to the perennialization of California grasslands, as noted in a previous study, where there's a loss of rare annual species that contribute to the high diversity in coastal grasslands. To summarize, we found that coastal grassland restoration was largely successful at reaching project-based goals and achieving a standard performance metric for native cover. These findings are exciting because both my advisors and I and the practitioners I've worked and talked to mostly thought grassland restoration was potentially being lost to invasive species. However, as indicated from plant surveys and management interviews, there are still major issues with invasive species management and restoration. Although projects were successful at reaching native cover goals and restoring some species richness, results indicate that the full complement of species regionally available are not always being restored. Most projects indicated that although they achieved their project-based goals, they would have ideally worked more on their project to increase the habitat quality if they had sufficient resources. We found that there's difficulty in restoring the full complement of species regionally available, which may be caused by several factors. Similar to what Lesage, Press, and Hull found with endangered species introductions, risk-averse practitioners may be more unlikely to work with particular species that are hard to collect because it'll be more straining on an already limited budget, but also because it makes it difficult to achieve project goals, which is also supported by our management interviews. Some annual species have specific disturbance germination cues or only do well on certain rain years, which may also cause the project not to reach project goals in certain years. And again, no one really wants to fail at reaching their project goals. Aside from a limited budget to source local materials, sometimes rare species only produce seeds in large amounts on certain years, making them further inaccessible. Accordingly, more research will be needed to be completed on rare species, life cycles, and nursery practices, because risk aversion and preferential species use could further contribute to biotic homogenization in future regional biodiversity loss. So now that we know how restoration projects have been successful and discussed with practitioners management limitations and tested methods for adapting restoration for future droughts, we can make some final conclusions. To summarize, my dissertation shows that coastal grassland restoration is largely successful at restoring native cover, but not as effective as in recovering biodiversity. Forecasted drought will have strong filtering effects on plant communities and may limit what types of plants or traits of plants will be able to establish. To summarize, the field experiment found that functional traits can inform plant selection and that key traits can help reduce mortality risks, thus improving establishment and restoration success. Trait development and trade-offs were driven primarily by abiotic stressors like drought and not as much by biotic stressors, indicating that selecting plants more so for drought adaptations instead of competition can potentially improve outcomes with forecasted climate change. Some species may also be more ideal for restoration, especially in invaded areas due to competitive release. However, regional vegetation surveys and management interviews indicate that restoration practitioners and land managers are already preferentially selecting species for restoration, resulting in regional biotic homogenization. Using suggested drought-adapted methods may further exacerbate biotic homogenization during restoration. So future work will need to focus on how to balance meeting restoration goals while increasing regional diversity. With that, I want to thank you all for attending my talk and listening today, and I'm happy to take any questions that anyone might have. And I also want to, again, thank especially Vicky Chung and Leslie Gorin, who made this amazing graphic and many other amazing artistic graphics that I've used to share out my research. Thank you.
Thanks, Justin, for a great talk. We have a policy in our department that we generally give the first question to a student. That could be a graduate or undergraduate student. So if there's a student that wants to either, it would be helpful if people raise their hands, we can do that, or if you can put it in the chat too. Either one, we're getting a lot of chats in the chat here. Justin congratulating you and telling you what a good job it is. There's somebody who would like to ask a question, a student.
While we're waiting, I'd like to note that we had a lot of people visiting from other departments on campus and other campuses around the state. And so I want to thank our visitors for being here as well.
Okay, I'm gonna give like one more minute for any student want to ask a question. Oh, well, I'm gonna go with, I don't know who Richard Brockman is, but because they have their hand, why don't you go ahead, Richard?
Hi, yeah, grad student from Cal State Northridge.
Great.
Good job, that was phenomenal. My question, so preventing this biotic homogenization seems like a pretty big deal. That it kind of seems like making vegetation monitoring that much more important on these landscapes. How do you think that we can incorporate nearby vegetation monitoring in habitats that, you know, maybe aren't being restored, but like have a lot of that native grassland? How can we incorporate that into seeds that these restoration folks can then use in their projects? Like, how do we bridge that gap between vegetation monitoring and actual grassland restoration? Thank you.
Yeah, thank you. That's a great question. I mean, unfortunately right now, one of the biggest pitfalls is that there isn't enough funding for most of this monitoring. And as I mentioned, most restoration projects are not really even able to monitor their own project after it's been implemented. So trying to put resources out there to monitoring nearby projects that could supply seeds would definitely be invaluable. But with current funding structures, I'm not sure if it would be possible. I think potentially partnering with agencies like the Rancho Ana Botanic Garden, which has some of this infrastructure and does do some kind of rare plant mapping already, could be a useful way to start. And then potentially trying to set up coordination, at least maybe within the region, to communicate between practitioners, between which species people are using and what species people are testing, and how we can also share kind of that that load as we go forward.
Okay, at this point, I'll open the questions from anybody. We don't have any so far in the chat. I think, Justin, yours was so clear, maybe that no other questions. Oh, okay, I'm gonna go with Edith as another student, and then we'll go to Anne after that. Go ahead, Edith.
Hi, thank you so much, Justin, for your talk. I would like to follow up with the idea of management strategies and the kind of the role of the humans in shaping the landscapes. And so with that, I guess I was wondering what strategies like have you actually like reflected on that could help bridge that science, you called it the science practice gap. I guess from your experience working with the restoration managers, and you've already like alluded to some of them, but I guess what do you think is really gonna push the needle forward for restoration?
Yeah, I mean, in terms of the science practice gap, I think one of the most important things is just to make sure that when you're planning restoration, like your research ideas, I know a lot of times when we're planning it, we have to think about theoretical concepts and what people will fund. But we also can go out and we can also go out and talk to the practitioners, especially in our local area, or to those that we think may be using some of these practices or the research you are hoping to generate that will hopefully lead to management implications, and ask them, well, is this something that you find is useful? Or is this something that maybe we kind of practitioners might know already from trial and error and communicating with other practitioners? And sometimes you can't hit every point, but you definitely should, I think, in my opinion, it's always best to do due diligence and go out and talk to people. And during your project, you can always revisit and kind of discuss what's happening and think about, well, how is this turning out, and what are my current results, and how can this potentially help management? And then you can go to them and ask them, do you think this is useful? And if they say no, well, then I guess maybe we should think a little bit more. But yeah, there is difficulty in balancing how you get funding and the type of work you do.
Okay, Anne, you want to go ahead?
Yes, congratulations, Justin, for the whole talk. I want to also pick up on that last part of your talk about your discovering this regional homogenization, which I agree from the little I know of this arena would be really important. So I'm wondering if, in addition to thinking about ways that you can kind of spread the word about that effectively to policymakers just to get attention to it, because I'm guessing there's not good understanding of that. Have you given any thought to particular state agencies or even key committees of the legislature to try to draw attention to this, to essentially help advance more funding for monitoring? It seems like, with the state's attention to climate resilience right now, this work and your insight is pretty timely. But there might actually be kind of incipient support for more funding, and you might be well positioned to really make the case for it.
Yeah, totally. Thank you for that great question. So I currently did work on a paper with my advisor, Karen, and thinking about biotic homogenization and potential policy recommendations at several scales of government. And so, I guess some of the takeaways from that is really thinking about basically housing a new type of unit within pre-existing government structures, potentially at the state level in like California Department of Fish and Wildlife, and as a unit that can kind of coordinate restoration activities or inventory, at least restoration activities that are happening across the state. And that way we can also at least have some type of regional coordination, which has infrastructure to provide support. And in terms of lobbying the legislature, I don't have experience doing that yet. I am on the board of California Native Grassland Association, and we've been working towards things like that, and I'm trying to get more experience in doing that. But if you have suggestions on how to work towards that, I'm happy to take some offline as well.
Maybe I'll follow up too. That is something that I would say Justin's is a very clear example of this biotic homogenization. We've discovered that this is something that happens globally in restoration, this tendency to use the same species over and over again. And so in this paper that we have in review, we've looked at different ways to maybe allow experimental designations, different ways to maybe design mandates and compliance so that it's a little more flexible to allow for some species that are harder to propagate. So let me go to Julie Larson.
Hey, Justin, can you hear me?
Yeah.
Congrats, Justin. That's an awesome talk. And I can't believe how well the diversity of your chapters together tell such an interesting story. I have a question that kind of spans all of them. And leading off the biotic homogenization question, so it sounds like a lot of people are using the same species, right? And I'm wondering, but I think it's really interesting that you were talking to all of these different managers, and I'm wondering if any of them have thought about trait-based science or trait-based restoration, or if they even know what trait-based restoration would look like or what it is. I think it's interesting and important because I think one of the things, correct me if I'm wrong, but I feel like one of the things that you found in the first project you discussed is that actually growing faster can be a hazard for survival, right? In which case the idea, to always choose plant species that grow really quickly might not always be the best choice, right? But that's something that managers are looking for, right? And I'm wondering where the gap is between trait-based science and restoration ecology in your mind. And if you have any thoughts or plans to say, if we need to diversify the species that we use, is one of the steps there to be able to study trait-based ecology in a wider range of potential restoration species so that we kind of know what their strategies are and how to use them? Like, is that even a possible path forward, or are there still barriers that you see there? Does that make sense?
Yeah, I think so. So I think there's multiple answers. So I'll just start with, I think that it is possible to use trait-based selections. And I do know some practitioners that are trying to work towards that, especially those that are generally with agencies with more funding. I find that sometimes government agencies and state and federal are able to think about these types of things. I find that other practitioners I speak to often may have heard of using trait-based selection, but it's more in terms of a broad sense. Like when I used to do restoration, we used to choose plants like that, or plants that could spread rhizominously or asexually. We didn't necessarily measure leaf traits, for example, or think about leaf traits, because that's just not really feasible for people on the ground because it takes so much time and there's not enough information for all of these species. I think if eventually the programs become more streamlined and I guess uniform, and there's more data provided, and which I think researchers can do that part, eventually it can be implemented at a kind of like at a, it can be uptaken more easily in a sense. I do think that's a really interesting point that you're bringing up about the balance between quick growing species to meet restoration goals and things like that. And I do think in some cases it's hard for practitioners and land managers and for us to really know whether or not these fast growing species are going to die later, because a lot of times projects don't have money to monitor after. So they want to meet their targets. And if that meets their targets the first year or the second year or up to the fifth year that they have to monitor, then that's great. And hopefully it sustains and we all want it to continue to work. But in reality, a lot of us realize that without additional work and funding, it may not be possible. And sorry, I'm not sure if that answered your question fully. So if there are additional points that you would like me to touch on, I'm happy to.
No, that's great. Thanks, Justin.
Okay, I'd like to go to Todd. Todd, you want to ask a question from U.S. Fish and Wildlife Service?
Sure, thanks. I was curious just what you thought about the success criteria and how appropriate that was, maybe like biologically or ecologically, for determining whether it's actually successful or not. So meeting a regulatory objective is one thing, but actually having biological significance could be another. So
sure.
That and the species richness.
Sure. So we have some data of remnant coastal grasslands in the area, and oftentimes they can sometimes achieve 40% native cover and sometimes even up to 80, but it's rare and it's very difficult. And my first thought is that it is a little, for me, I think it would be a little unreasonable to expect practitioners with limited budgets to achieve the same level as remnant systems, which we know will require a lot more work. In terms of selecting 25% native cover, and which I know may seem low to some people, especially those that work in other systems, that is supported both by compliance monitoring and by how we classify grasslands, which classification of grasslands only require 10% native cover, And the main reason we chose to use 25% native cover is, or the reason it might seem low compared to other systems is because in forest, for example, after the trees grow, they have very strong biotic filtering through shading. And in wetlands, for example, there's really strong abiotic filtering through flooding. And so that limits some of the invasion. In grasslands, they're completely open and they're regularly disturbed. And so that really allows for a real serious problem with species invasion. And so I think eventually one day we can aim for higher, but I think unless we can allocate more funding for restoration, it would be a little unreasonable to necessarily ask practitioners to achieve something like 50, 80, 100% cover. Because in my opinion, that would just be setting up people for success. And setting people up for failure, and then basically kind of causing a positive feedback cycle where people think restoration is not successful, and so we shouldn't further fund it. I'm not sure if that helps.
So we have, Todd, do you have a response? We have,
thank you.
We have several questions in the chat. Somebody said, Brett, I would like a link to your work. There was a question from Jim Belzy about whether your work would relate to how it might correspond to inland grasslands in California. Do you have any thoughts on that?
Yeah, so I expect there to be similar. I expect there to be similar issues. I'm not exactly sure which portion you're referring to, but if you're referring to biotic homogenization, I expect it to be similar and potentially even intensified. Because there's harsher environmental conditions in Central California than coastal California, which could potentially limit the species that do well. And so that's what that would be my initial thought on it. And in terms,
I'm gonna try to go for one more question here from the chat. I'm gonna go with Gray Hayes' question. Isn't self-sustaining a factor in determining success? What if they have plantings that live for a while, but they don't reproduce?
That's a great question. I'm actually trying to look at that right now with some senior thesis students. We collected soils and yeah, so I'm really concerned with that. I do see that certain times we are achieving native cover, but I'm wondering what's happening with the seed bank and are they going to regenerate after these die? And so the soils I collected from 2019, I'm working with some students to grow out up at the greenhouse to basically assess the relative proportion of native and non-native species germinating and really think about, well, are these restoration projects also sustaining past the cover and vegetation we put there?
Yeah, maybe we can say that the two students, Alyssa Angelin and Nick Mazzotti, are with us here today. So we'll be looking forward to their results. And we are at time. I see some more questions, and Justin has kindly said, I know some people need to go and the faculty need to go off faculty meeting, but that I want to say thanks again, Justin, for an excellent talk, very clear and thorough. And that if anybody wants to stay on and ask him questions, or you can email Justin, put your email in the chat, to follow up with that and to get announcements of his upcoming papers that are coming out. So if you're in the process. So anyways, congratulations, Justin. We're really proud of you.
Thank you. And I do keep a list, so yeah. If anyone wants to follow updates, send me an email and I'll put you on my list. And I regularly send out updates to practitioners and others.
You can also follow him on Twitter. He tweets about it a lot too. So